Are we live? Okay. All right. Yeah, it's live streaming straight to YouTube right now. Okay. Also, I'm going to hit record here in case there's some fail. So here we go. Hi, this is Miss Litton, and this is Honors Bio Unit 6 review. This is my wonderful 7th period group. Say hi. Hi. All right. So on this one, and hopefully you got a chance to already look over this review from when I put it out before. Somebody check to make sure it's presenting right, but don't put up the volume because then it'll be annoying. Is it okay? And can you read the words forward, not backwards? Okay, good, because for me it's backwards. All right. So chapter 20. Just a moment. Chapter 24 was all about DNA biology. Now, remember, we want to be able to tell the history, those key experiments that we talked about, right? Start thinking of some key experiments in your head. Say my name, say my name. What are some of the experiments? Hershey and Chase, I love it. What else? Rosalind Franklin and Maurice Wilkins. Who else? Chargaff. Give me another name. Watson and Crick. Is that it? All right, so you want to be able to differentiate between what they did and what that contributed. I'll go over that with you. Then DNA structure. So remember that DNA is a type of nucleic acid, right? And so all nucleic acids are built out of nucleotides. And a nucleotide consists of what? 3 things? Base, sugar, and phosphate. And you want to be able to differentiate between a DNA nucleotide and a RNA nucleotide, okay? And know the arrangement of that. And I'll review that with you too. Make sure to note anything like, please, Miss Litton, go over this because I already reviewed and I'm not sure. DNA replication. You are going to have different questions in each class, as you know. That would be something I would ask is like, what are the enzymes and steps of DNA replication? What are the steps of transcription? What are the steps of translation? Or I would show you a picture of something getting translated and ask you to identify the different items on there, like codon versus anticodon. Does all of that sound familiar? Okay. So, so transcription is DNA to RNA, all kinds of RNA. What are the 3 kinds of RNA? mRNA, tRNA, and rRNA, right? And we would want to know what each of their jobs were. Transcription is the formation of any kind of RNA. And then translation is using that RNA in order to build proteins. Now, what do proteins consist of? Amino acids, so a polypeptide chain, good. Then we're going to talk about mutations, and we talked about the different kinds of mutations. You can have a point mutation or a what? Frame shift. Which one's worse, do you think?
Frame shift.
Frame shift is always bad, no matter what. Because a point mutation, you could have a what kind of mutation? Silent mutation, right? Okay, so that is chapter 24. So the first one was Hershey and Chase, right? Remember, you'd always chase down a Hershey bar. And the big thing there is they were trying to find out, is DNA our transforming agent? So they labeled the 2 components you would find inside of a nucleus. What are the 2 things you would find inside of a nucleus? DNA and proteins, right? And so what they were they did not know what a bacteriophage looked like, remember, but they labeled the DNA with an isotope of phosphorus, which would label all the phosphate, right? And they labeled the protein with S35. So they used that, and then they looked to see what got shot into the bacteria. Because remember that bacteriophage is like a lunar lander. It goes like this, and it shoots in its nucleic acid. And so we know that now. They didn't know that at the time. And so they were looking to see what got shot into the bacteria. And sure enough, it was radioactive phosphorus. What was an unusual tool that they used that you might use every day?
Blender.
A blender. And what did they use the blender for?
To separate the...
Yes. The bacteriophage from the bacteria itself, right? So that was one key experiment. Then we talked about kind of the overview about DNA. We have 46 chromosomes, 2 meters of DNA. It's very, very tiny. There's about 30,000 genes on our DNA, right? A gene is a series of nucleotides. But there was somebody who did an experiment looking at adenines, thymines, guanines, and cytosine. Who was that? Chargaff. And what he found is when you look at all different species, you can have various numbers of each of those nitrogenous bases. But what was the pattern that he saw?
A equals T, G equals C.
Yeah. He didn't know who was binding, but the adenine numbers always equaled the thymine, and the guanine always Cytosine. Now, you could have one species that had 20% adenine and another species have, 40% adenine, right? But however many adenines it had the same percentage of thymines and guanines with cytosines, right? Okay. And then that makes me think about purines and pyrimidines, because you always have a purine binding with a pyrimidine. Good. All right. And then Rosalind Franklin. Do you remember she was using X-rays, right? X-ray diffraction and shooting X-rays through crystallized DNA. And remember from that, she discovered that it had to be a what shape? And it was the constant diameter across the top. So who used all of that information? Watson and Crick. So Watson and Crick came up with the structure of being a double helix. If I were to give you a series of bases, like let's say I covered up this side and I just said TCG, what would be across from the T? A. And across from the C? G. And across from the G? C. You would be able to recognize if there was a mutation, right? If across from this T there was a G over here, still a purine, is that okay? No, okay? Because adenine always binds with thymine and guanine always binds with cytosine. What holds those bases together? Hydrogen bonds, right? And who else are they bound to? They're also bound to what? Sugars. And who are the sugars bound to? Phosphates. Okay? So it's a double helix. If I were a fireman and sliding down the outside of that ladder, what would I be grabbing onto? Alternating what? Phosphate sugars. If I was climbing the rungs of the ladder, what would I be grabbing onto? Nitrogenous bases. Right? Are you okay with that? All right. So this is just a refresher on purines and pyrimidines, but I think purines are 2 rings, pyrimidines are 1 ring. Okay? All right? And if I were going to draw this accurately, how many hydrogen bonds? One of these has 1 more. Which one is it? G and C has 3 hydrogen bonds between them. Okay with that? Tell me, I'm going to just jump back because I don't have a slide for it, and I don't know why I don't have a slide for it, but what's the name? Okay. What's the name of that sugar right there? Deoxyribose. If I was showing you RNA, what would be the name of that sugar? It would be ribose. And whereas DNA is double-stranded, RNA is single-stranded. Where DNA has the base thymine, wherever you'd want to put a complementary thymine, instead you would put a complementary uracil. Okay, good. All right. Ask your bio buddy, do you have any questions on that so far?
Do you have any questions so far?
Okay, anything you want to ask me before we leave that? Just the structure and history of DNA. Are you good? I move forward? All right. So when we talk about DNA replication, we describe it as being semi-conservative because each old strand serves as a template for a new strand. Good. And if I give you any series of bases on the DNA, you could tell me the complementary DNA from that. You could tell me the mRNA if I was doing transcription. You could tell me the tRNA, right? And if you had a chart, you could look up the amino acid it codes for. Yes? Yes. All you need is one of them. It doesn't matter which one I gave you. If I gave you the mRNA, you could work backwards to tell me the DNA. You could work forward to tell me the tRNA. And you could look on the chart to tell me the amino acid. Yes? All you need is one, and you can tell me everything else, right? Okay, so on DNA, differences between DNA and RNA, check that you understand that. Ask your bio buddy, are you strong on that?
Yes. It doesn't turn into it, it's replaced by DNA, deoxyribose, RNA, or ribose.
All right. Why don't, because I didn't have another slide for it other than this one. Let's review DNA replication. You ready? Here we go. We're here. Right, we're here. Okay, so topoisomerase undoes the supercoiling, and then we unwind and unzip helicase. We have a leading strand and a lagging. Okay, pause right there. If this is the leading strand, what end is that? 3 or 5? 3. And that's because I'm counting the carbons when I go around the sugar, right? Okay, so if this is 3, then my elbow is 5, and then across the street from it, this elbow is 5, and this is 3. So this is my leading and my lagging strand. I'm going to use DNA polymerase 3, and it's going to go boom, boom, boom. If this is A, it's bringing in a T, G, C, T, okay? And it can do it continuously on the leading strand because it reads from 3 to 5, right? But on the lagging strand, it's a little bit slower. We go boom, boom, boom. And we're doing that because it's backstitching, right? And what do you call those fragments? Okazaki fragments. They're really 1,000 to 2,000 base pairs long. If you're in AP, we would first lay down primer with RNA primase. Okay, I just had to put that in there. Okay, then we would use, in AP, you would use DNA polymerase 1 to remove the primer. Then you would use what to seal all the fragments together? Ligase. Then these will twist up and they would form sister chromatids held together by a centromere, separate during anaphase. Anaphase of what? Anaphase of mitosis. Good. All right. So you would do that where in the cell cycle? In the M stage, okay? When would you do the DNA replication? S stage, right? What follows the S stage? G2, then M, and then G1. Yeah, cycle. Remember your cycle? Do you mean to draw it or are you good? It's not on your test, but I just don't want you to be confused.
What's not on?
Draw it?
Wait, what's not? It's just review.
Remember in the cell cycle, remember this? You had the M stage, and then this whole thing was interphase. Remember that? And interphase was broken down into what? G1, S, and G2. And then mitosis, remember prophase, metaphase, anaphase, telophase, and overlapping telophase was cytokinesis. You got that? All right. All right, so now let's take a look at this picture. What this is showing you is you copy all the DNA, NY, all of that, boom, if you're getting ready to make another whole cell, right? Because you've got to replicate the entire code if you're getting ready to make another entire cell. But if a cell is just going about its business and it needs proteins, and remember the DNA codes for those proteins, that process of taking the DNA information and get the specific section of that DNA information that you need, that you're going to make mRNA, right? That is the process of transcription. And then you're going to use that mRNA, if you're in a eukaryotic cell, out on a ribosome, and you're going to translate that into a polypeptide chain. So transcription is using DNA to make what? RNA. Translation is using RNA to make a protein. Okay, so that would be known as the central dogma, right? So if I did something like this, okay, oh, here. What would this be called right here? What would A be called? Transcription. What would this be called? Semi-conservative replication. What would this be called? Translation. Where would this take place? On the ribosome, right? That would take place on the ribosome. Where would this take place? In the nucleus. Where would this take place? In the nucleus. Yes. Shh, I can't hear. I can't hear. Shh, go.
Is the rRNA the actual ribosome?
Okay, so when you do translation, you're going to use the mRNA, right? So the mRNA, okay, so here's your mRNA. I'm going to answer your question. It's going to get there. What do you call every 3 bases on the mRNA? Though they're not spaced like this, they're spaced evenly, but what would you call every 3 bases? This would be a codon. Okay, so your mRNA got transcribed from your DNA, yes? It's sitting on a ribosome. Technically, there's a small and a large ribosomal subunit. What is building this ribosome? rRNA and some proteins. rRNA and some proteins is what that is. And then you would have your tRNA. What is this right here? Anticodon. What's this? Amino acids. So the tRNA, the mRNA, the rRNA all got transcribed in the nucleus in a eukaryotic cell. But they all serve different purposes. It's only the mRNA that is translated into a protein. The tRNA is a worker and the rRNA is a worker. Does that make sense to you?
Is rRNA still like code that it matches up with, or it doesn't?
No. rRNA is just a nucleic, you know, like if I said, what is a fat built out of? Tell me, you guys, what is a fat built out of? Oh my gosh. 1 glycerol and 3 long fatty acid chains. And then you call that was a big pull. That's a big pull. They're super smart though. Don't let them fool you. Okay. That is just, I'm giving them a chance. That is just what you would call a fat, right? It's a fat. This is a ribosome. What builds a ribosome? rRNA and protein. So you don't talk about codons on rRNA because it doesn't have any. The only thing that has codons is your mRNA. It's the only thing that has codons. Does that make sense? Yeah. Okay, yes?
Does the small and large unit face the nucleus? This is just...
No, it's not a cis-trans thing.
Oh, okay.
Yeah, it's in between. Yeah. All right, so now let me go back to this, right? So transcription is any kind of making of RNA. Translation is building a protein using the 3 RNAs. What's the M stand for? Messenger. It has the message. That message is in 3-letter words, right? Codons. Those are complementary to anticodons on the tRNA, right? And mRNA is also complementary to something else besides tRNA. What else is it complementary to? The DNA that made it, right? Those are all complementary to each other, all right? Let's see. So let's talk about, let's do the transcription song. Ready? Okay. Remember when we did replication, we were replicating all the chromosomes, right? Whether we were a prokaryote and it was a single circular chromosome or all the linear chromosomes. Transcription is specific DNA. Remember later in the next chapter, we talked about euchromatin and heterochromatin. Do you remember that? Heterochromatin is tightly coiled and put away. It's not going to get transcribed. Euchromatin, you can transcribe that. It is available for transcription. Does that make sense? I'm just trying to link those two together, okay? So the DNA unwinds, unzips. What are we using? Helicase. We got a leading. What are we going to do with this one? Put the lagging one away. So now we're not going to use DNA polymerase. What are we going to use? RNA polymerase binds to the promoter. That's the beginning in a prokaryotic cell of a whole unit. Starts with an O. Do you remember what you call that whole thing that where the control? Yes, on operon. Boom. Okay. So it's the controlling regions and the structural genes. Okay. One more time. RNA polymerase binds to the promoter. Then you boom, boom, boom. And if it's an A, you're going to bring in a U nucleotide. It's still going to have also a sugar connected to it. What's the name of that sugar? Ribose. And it's going to have a phosphate. Phosphates are the same, right? So if it's an A, you bring in a U nucleotide. G, C, G, T, A. You still have A's, right? Make your mRNA. Peel it off. Set it aside. Go get that lagging strand. Zip it back up. Put your DNA away. Now focus on your mRNA. Put on a cap, 7-methylguanosine, and a tail. And the tail is 100 to 200 adenines. And then you cut out the introns before it leaves the nucleus. So you cut out the introns, what's left behind? Exons, because the exons are expressed. Then your mRNA goes out on your ribosome. Every 3 bases on the mRNA is called a codon, which is complementary to a anticodon on the tRNA. What does the T stand for? Transfer. What is it transferring? Amino acids. So tRNA1 lands, tRNA2 lands, tRNA1 dumps his amino acid on to tRNA2, and then he leaves, and this keeps going again and again and again, and you build a polypeptide. Now, is that polypeptide done? No. That was translation. What do you have to do post-translation? Fold it into the right shape. So it's a functional protein. That's post-translation. Okay? Yes?
Sorry, what's the cap in the tail?
The cap is 7-methylguanosine. You don't have to memorize that.
Okay.
And a polyetail. I just can't. It's hard for me to not say it. Your practice doesn't start yet. Not till 3:30. I'm just saying. No, it doesn't take you 35 minutes to walk down. All right, so what we know is every 3 bases are a codon. We know how to read that chart. There, you could look up videos, plus I have videos for you located on the calendar that you could watch, so you could re-watch some of those examples. We talked about mutations. We said errors in this part, boom, 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 that is rare, okay? But you can be exposed to mutagens like radiation of some sort, a chemical, smoking, right? The sun. Those could cause mutations. And we talked about transposomes, jumping genes, right? If it's just an error in one nitrogenous base, that is called a what kind of mutation? Point mutation. But if it throws off your reading frame, this was our example, the big fat cat ate the big rat or whatever I put right there, okay? And if you eliminate one entirely, okay, then it throws the whole reading frame off, so it will not make any more sense, right? So that's a frame shift when all of them are wrecked. If you just ruin one codon, that's called a point mutation. Like if you have a G where you should have a C. Sometimes point mutations don't cause any problem at all because when we look at that codon chart, sometimes there's multiple ways to get the same amino acid, right? Yes?
What would happen to the just 2 instead of 3 so it's no longer a codon at the end?
This one, probably you wouldn't have a tRNA bind, so it'd almost be like a nonsense mutation at that point, but all these other ones are already screwed you up. Yeah, you already have bigger problems. All right, that is it on, and then, transposomes were jumping genes. Okay, so that's it on that chapter. Now, in that chapter, because somebody brought this up, they were doing practice questions at the back. We do our biotechnology, not the next unit, but the unit after. So I'll revisit this chapter for the biotech part for you guys when we are ready to do that biotech lab. So don't worry about anything with biotech on that, okay? All right, so this is the latest chapter, chapter 25, control of gene expression. And we focused on the operon, right? And, let me give you all your topics, okay? So these are all things relating here in blue to the operon, right? And then we talked about the difference, also in eukaryotes as well. Then we talked about the differences. We talked about cancer between proto-oncogenes and tumor suppressor genes. We talked about the causes of cancer to diagnose cancer and characteristics of cancer. Right? Go. All right. So in a eukaryotic cell, when we looked at levels of control, you want to be able to differentiate the levels of control, and then we'll go back and talk about a prokaryotic cell. If your DNA is tightly coiled and put away, then you're talking about chromatin structure. Your best example for that was bar bodies. Do you remember what bar bodies were? Yeah, the second X in females. Okay, condenses, right? So if you are a biological male, XY, you would or would not have a bar body? Would not. You would not have a bar body. But if you are a female or Klinefelter male, XXY or your XX, you would have a bar body. It would condense and you can see it right in the cell. That's an example of chromatin structure. And we looked at the calico cat, right? The female calico cats have those mottled appearance because you don't know which X gene is going to be expressed for color. We also looked at bar bodies on our hyperdoc with identical twins. Remember, one of the twins was colorblind and one wasn't. Is that all coming back to you? You're quiet, yes? Okay. Transcriptional control, who's missing an L? Transcriptional control, this has anything to do with RNA polymerase, what? Binds to the promoter. Anything supporting or inhibiting that is transcriptional control. Post-transcriptional control is once you've made your mRNA, you've got to protect it on either end. If you're a eukaryote, a cap and a tail, and you're cutting out what? Introns. So different genes are expressed. So that's post-transcriptional control. Translational control is anything to do with mRNA being on the ribosome, and anything interfering with the tRNA anticodons being complementary with the mRNA codons. Post-translational control is that folding we talked about that happens afterwards. Chaperone proteins fold that protein up. Yes?
If there's an error at one of these controls, does apoptosis occur, or do they try to fix the error?
Well, it depends. It might be just a disease that you have. If it's in the DNA, if there's a problem with the DNA, hopefully you hope apoptosis occurs. Okay? That's why you have P53 genes. That's why you have tumor suppressor genes. They try to stop it. But if you have a problem with your protein, it's more than likely a problem with your DNA in some way. Okay? All right. Now, when we talked about the operon, okay, remember this is in prokaryotic cells. They do not have a nuclear envelope. Where they do transcription is where they do translation, just right there in the same place. Literally, as soon as they make the mRNA, as soon as they're building it, it can start getting translated right after you make it. Their DNA is controlled via operons, and we look to see what is an operon. An operon is your promoter, right? And who likes to bind to the promoter? RNA polymerase. The operator, who likes to bind to the operator? The repressor, and then here are your structural genes. Remember, this means farther upstream. Farther upstream is where you have your regulator gene, and you code for, in this case, an active repressor. Just so you know, you could also code for an inactive repressor that has to be turned on. This is an active repressor that has to be turned off, right? And so we looked at, oh, I didn't do that one picture, but that's okay. Okay, so here's your repressor, it's on the operator, and here's your RNA polymerase. If RNA polymerase is trying to get on the promoter, it can't if a repressor is on the operator. We looked at this diagram right here. But is that a bad thing that RNA polymerase cannot get on?
No.
No. It's not necessarily a bad thing, it's because the products of these genes are not needed. So if they're not needed, it's fine if you're not transcribing them. In the example we gave of the lac operon, what would turn on the genes? Lactose itself, right? Lactose was the inducer that would bind to the repressor and pull the repressor off the operator. Now RNA polymerase can get on the promoter, and that's great because when you transcribe these structural genes, they make enzymes to digest lactose. So it's a nice way to control. Remember we talked about right now in your bedroom, your lights should be off. Is it okay that they're off? Yeah. You don't want them on. It's wasteful if they're on, right? But when you are present, when lactose shows up to a cell, now you need these enzymes. So when you're present, you flip the switch. Lactose goes over and binds to the repressor and flips the switch, pulls the repressor off, so RNA polymerase can bind to the promoter and you can get those genes transcribed. Okay? Ask your bio buddy, how are you feeling about the lac operon? Yeah, that's just one example, and that was in bacteria. Yeah. Okay, now in eukaryotes, we did talk about that instead of so much about repressors saying no, we have transcription factors that say yes, that facilitate transcription. And we talked about transcription activators binding to an enhancer region, which then binds to a transcription factor, right, to help RNA polymerase bind to that promoter. Okay, we already mentioned euchromatin and heterochromatin, right? Euchromatin, you can transcribe that. It is available. Heterochromatin is dark staining, not available for transcription. An example of that were bar bodies. We've talked about the operon for transcriptional control, post-transcriptional control, cap tail. We talked about translational control, anything that gets in the way of the ribosome, and then post-translational was the folding. Okay, then the last big topic of this unit was about cancer, and one of those would be about differentiating between cancerous cells and non-cancerous cells. They physically look different, okay? So can you ping pong back and forth with whoever you're sitting next to? Whoever's in the blue seat, you are normal. Slate, you've got cancer. Okay, back and forth. Go real quick.
So cancer.
All right. Now, the big problem with cancer cells, okay, they defy normal regulation. Okay, normally there has to be a signal, let's say, to move the cell cycle forward, and it just defies that. And they have the ability to metastasize, to spread to other areas. And that spreading is bad because, remember, when it spreads to these other areas, then they set up camp, and then the cells in that particular area can't do their job, right? And you have too many of these cancerous cells. We talked about the term angiogenesis, where normally if you had this large grouping of cells, you don't have the capillaries to feed that cancer tissue, that tumor, right? And so it won't spread because of that. But if they can get blood vessels to grow towards the tumor, that's called angiogenesis. And then when that happens, they have all the nutrients they need, and they may acquire that mutation to like go through the basal lamina and spread to other areas, right? And that's when we say it stage 1, stage 2, stage 3, stage 4. We talked about in testicular cancer, stage 1 is when you have cancer of the testes. Stage 2 is when it moves to your what? Do you remember? Lymph nodes. Stage 3 is when it moves to your lungs, and stage 4 is when it moves to your brain. Did I not talk about Lance Armstrong with you guys? Oh, no. Okay, well, now I just taught it to you. Okay. It's okay. Don't stress out about it. There's not a question about stage 1, 2, 3, 4. That was just to give you an example of the cancers have real typical organs they go to in their sequence. Yes?
On your computer, it's the software.
Oh, did it just do that?
I don't know. I just saw it.
Uh-oh. Close.
It's still going.
Yeah, yes. That is YouTube. I did a fail-safe, so I'm recording it using Doceri to load in case for some reason this failed and it didn't load.
You can check on YouTube how long it's been there, right?
I could. Yes, I could. I think we're good. Yes.
So during angiogenesis, is this
Yes.
where it kind of becomes its own part of the body and has all the nutrients going?
Well, all angiogenesis is, by definition, is when a cancerous mass gets blood vessels to grow towards it and supply it with nutrients and oxygen and a way to remove its waste and carbon dioxide. Yeah, that's all angiogenesis is. Yes?
Can the tumor or like the cancer metastasize if it doesn't have angiogenesis?
Well, it depends on where it is located and where the tumor goes. If it has easy access, let's say it's spreading like breast cancer, and it can get into a lymphatic vessel, it's good. Because now it can spread to where and take whatever nutrients of whatever location it's in. Yeah, you don't want to get, that's the big thing. You don't want it in your lymphatic vessels because that's a whole train railway system to your entire body, just like your circulatory system. Okay, so let's finish up. There's 2 more things here, and those were those 2 types of genes. So proto-oncogenes, they are not bad. It's just that they're in a position of what? What are they in? Power, a position of control, because they facilitate the cell cycle. So if they go bad, just like if an army general goes bad because they control so many other things, then it's called an oncogene. And oncogenes basically promote the cell cycle. They tell the cell cycle to go forward even when it shouldn't. So an example of that is the RAS gene, because it's in a position of power. And that's why I say RAS is like stepping on the gas. So you remember, it's like, remember that analogy I gave you? You're driving with your friend towards an intersection and you see like a stoplight up there. And so all cell signals would say to what? Stop. But your friend's like, yeah, and steps on the gas. And you're like, what are you doing? It's moving forward in the cell cycle. So normally RAS is not activated unless a what? Receptor is triggered by some growth factor. The receptor is triggered, it triggers RAS. RAS tells the cell through signal transduction and some transcription factors, hey, let's move forward in the cell cycle. Let's keep making more. But this is an altered RAS, a bad RAS. Okay, so he doesn't need this growth factor to trigger the receptor. He just says go forward in the cell cycle. Does this guarantee that I'm going to have cancer? No, because who would normally take care of a cell that has a bad RAS gene?
Tumor suppressor genes.
Tumor suppressor genes. So tumor suppressor genes, they are like the sniper, right? They're to stop the out of control cell. So tumor suppressor genes, like a very common one is the P53 gene. And this codes for what? Apoptosis. And technically it doesn't just say, oh, bad cell, okay? What it will do is it'll stop the cell cycle, try to fix the DNA. If it can't fix the DNA, then it takes the cell out, okay? And so if this would be in the analogy when you're driving your car, your friend's stepping on the gas, you're headed towards the intersection, headed for a crash. Normally the tumor suppressor genes would be like stepping on the what? Brakes. And so you're not going to wreck. But if your brakes what? Fail. Yeah, if they're broken, then you're going to still crash. So it's a combination of both of those for you in order to have cancer. So this was the P53 gene, stopping and checking the cells to make sure they're all right. Now that will happen regardless if it's a cancer cell or a normal cell, because just like to get on an airplane, like if you were wearing like things that say, I like to shoot up airplanes, I carry all bombs, you know, that's an obvious person that TSA is going to stop, right? Okay, if they're like strapped, you know, they're going to obviously stop them. But TSA checks who? TSA checks what? All cells, yeah. So same thing here. P53 checks all cells to see if there's a problem. Okay, it's just that if they let somebody through TSA that really did have a bomb. Does that make sense? Okay, and then last but not least, we said, how could you inherit it, or how could you get cancer? You could inherit it. It could be one that is passed on, like the breast cancer gene, right? And so you could check for that. And then it could be also due to your environment as well, because of what you expose your body to. Okay, I think we did it perfectly. Are there any questions you want to ask me before we go? You're smart. I love you. Make good choices. If you're reviewing this at home, don't stay up too late. I want you to get a lot of rest. You're ready to go for your test. And by the way, I know tomorrow is a shorter day for third period, but that period is longer. Third period students, darn it, fourth period students, be ready to go right at the start, because literally the bell's ringing, or I might even start you before the bell rings and get you at your group. So be ready to go.
For today, they dismissed like 15 minutes early. They had to go out.
Right. And we have a 12-minute differential. So, you know, it's only 3 minutes we got to make up. I can find 3 minutes.
Oh, yeah, I was gonna
Okay.
say, if we could stay in at lunch, but then we don't.
Okay, I hope that was helpful. Okay, bye. You're super smart. I believe in you. Okay, are you sure you want to stop streaming? Yes.